Okay, everybody, so we're getting out of chapter 16, which was essentially just the discovery of DNA, kind of how it was done. All right. And once DNA is discovered, that opens up a lot of understanding of how the molecule is used. So, naturally, one of the most important things to biology is transcription and translation. So, here's a big picture of transcription and translation. All right. I mean, you got to really, really look at this illustration. It's a great illustration that you want to look at and just kind of make sure you understand. So you can see here's the DNA, all right? Here comes the RNA molecule, and it is going to be moving and building the DNA molecule in the 5' to 3' direction. Okay, so you'll see here's the 5', the new 5' end of the mRNA, and you've got this whole, what we call the transcript, done in the nucleus. So transcription is done in the nucleus. And then you're going to build what's called the pre-mRNA. That's this guy. And the pre-mRNA has a bunch of non-coding stuff in there. And we'll get into that a little bit more later, what that means. But those are called introns, and there are exons in there. So the next thing that happens is that sucker has to be spliced. Now we're only talking about eukaryotes that do this. Okay, eukaryotes are the only ones who splice their mRNA. That means remove these non-coding lighter version, light pinky introns, and splice together the exons. It's just like cutting bad sections of a wire out, splicing the good sections together. Once that all happens, all that editing is done. It's done by spliceosomes, which are other proteins and stuff like that. That mRNA is going to leave the nucleus through the nuclear pores. DNA can't fit out those because it's double-stranded. This can't, it's single-stranded. And that mRNA is going to travel out into the cytoplasm where it's going to hook up with a ribosome. And at the ribosome is going to read the mRNA from the 5' to the 3' position. Everything reads. And reads and works that way. Okay, all the ribosome is, guys, is a complex made of protein and enzymes. It's an enzyme, and its job is to make peptide bonds. So at the mRNA, each codon will be read, and the appropriate tRNA with the right anticodon on the bottom is bringing the appropriate amino acids. So what happens is this is where the tRNA comes to the mRNA. It brings its kind of lands on the mRNA temporarily, and then a peptide bond is made right here, and then the E portion of that ribosome is where it's going to exit and go off and get recharged. And that means an enzyme will attach a new amino acid to a specific type of tRNA, and the process goes on and on and on and on. Okay, these mRNAs are not just read once. They're read many times, and that's why you see many ribosomes here, because they're gonna basically, one mRNA can probably make thousands and thousands and thousands of copies of the same protein. All right, so that's the big picture. And what we do in chapter 17 is we kind of break it down. We break down how the theory was developed a little bit. We're going to go through that very quickly, and then we break down all the steps of transcription translation. Okay, like how the enzyme hooks up with the DNA, how it makes RNA, and how it's edited, and then how it's read. A lot of stuff, but we're going to go through it pretty quickly. Notice the shape of this enzyme right here. Okay, that's the international symbol for RNA polymerase. So there's DNA polymerase that builds DNA. There's RNA polymerase that builds RNA. All right, and they all go, remember, favorite band. All right, one direction. They all go in the same one direction. So the transcript or the template is 3' to 5', and they build 5' to 3'. They all build 5' to 3'. Okay, as long as you remember one direction, you're going to be cool. All right. Now, the central dogma of biology is DNA is used to make RNA, which is used to make protein, or just gene to protein. So early on, back in the early 1900s, remember they thought that proteins were really the transforming agent. They thought that proteins carried all the code, and they were wrong. So there's this German guy named Archibald Gerard, and he was studying people who had L-captonuria, and that was definitely a black maple syrup smelling urine. Talked about that in class before. And basically he figured out that that these people were lacking an enzyme. So he felt that genes code for enzymes. Well, they can code for more than just enzymes, right? They can code for proteins, and some proteins are enzymes. But he was onto something there. Beadle and Tatum, they work with Drosophila. But really, a tremendous, the Nobel Prize winners here was 1958. This is shortly after the time period of Watson and Crick when Beadle and Tatum were working with neurospora. Now, neurospora is just good old bread mold, that blue stuff you see on bread. All right. And here's what they were doing. Kind of interesting here. So this is the wild type of neurospora, which is a fungus, and they were giving it minimal medium. That means like minimal nutrients. Very, very small amount of nutrient. Only the wild type survived. These mutants here, they were bombarding neurospora with X-rays and stuff like that, X-ray. And so when they bombarded the mutants were made by bombarding with a specific amount of radiation, basically. These guys, as you can see, with minimal medium only, would not survive. There's nothing in there, okay? That orangey line right there means there's life, all right? And then the next level of testing they did minimal medium, and they added ornithine, which is a precursor to arginine. Okay, it's an amino acid, it's a precursor. So when they added ornithine, of course your wild type survived because they can survive just on minimal medium. But when they gave it ornithine, the class 1 mutants also survived, but these two did not. When they gave it citrulline, then not only did the wild type survive, but so did the class 1s and the class 2s. And then when they gave it arginine, the final thing, they all survived. So from that, they're actually able to figure out that there were enzymes that were required to build one from the other. It's an anabolic process. Oops, anabolic process. So here's my precursor, and then an enzyme is used to convert that precursor to ornithine. And then another enzyme is used to build citrulline and arginine. So when they were mutating these guys, when they were bombarding them with X-ray, they were knocking out these enzymes right here, okay? So the class 3 mutants were the ones that needed the most. So they had all 3 enzymes knocked out. And the class 2 mutants, since they needed, as you can see, citrulline, they were getting enzyme A and B knocked out. But they could survive. If you gave them this, they had enzyme C to convert it into arginine. And the class 1 mutants, they were knocking out enzyme, let's see, they needed ornithine. So they were knocking out, I believe they were knocking out enzyme A. So if you gave them ornithine, they had enzyme B to convert it to citrulline, and they had enzyme C that can convert it to arginine. So, oh, it even shows you right here. Look at that later. Okay, so there you go. All right. Very neat stuff that they were working with. Okay, so transcription, translation, just remember the differences between RNA and DNA. I think by now you've heard it a million times. Okay, transcription is just the synthesis of RNA under the direction of DNA. Most importantly, it occurs in the nucleus of eukaryotes. Okay, in prokaryotes, bacteria, they don't have a nucleus. Just happens right there in the cytoplasm. Translation is the synthesis of polypeptide synthesis of a protein under the direction of RNA. Occurs at the ribosome in both prokaryotes and eukaryotes, and occurs in the cytoplasm, of course. Okay, both transcription and translation are unique to all life. Everybody does it. All right, it's faster in prokaryotes. It does not occur in stages. There's no editing, for example. That's the big, big difference. They don't edit their RNA. They make it, they read it. We make it, we edit it, meaning take out those introns, splice together those exons. But prokaryotes are very quick. In eukaryotes, it's called the primary transcript. That is the unedited version. That's the first version that's made. It has to be edited. That's the biggest difference. You're going to hear that over and over and over. Okay? The genetic code, remember there's 4 nitrogenous bases. So how many amino acids are coded for? So if we just coded 1 letter, there'd only be 4 amino acids. If we only coded 2 letters, then there'd be 16 amino acids because you got to think of all the combinations. 4 squared is 16. But there's 3 letters. So 4 cubed is 64. But there's not 64 amino acids. There's only 20. And what that means is that the code, when they saw this, oh, there's Marley barking in the background. When they saw this, they would say that the code is redundant but not ambiguous. So what that means is many, many codons code for the same amino acid. Okay, you don't for an amino acid to be coded, there's many different versions of it. You could have like UUU, UAU, all that kind of stuff. For example, Mr. Leucine. All right, leucine, Mr. Luce, I always make fun of them, but there's a lot for leucine, okay? And then to be not ambiguous means it never changes, all right? It's always going to be the same, never changes. UUU is always phenylalanine, there you go. Okay, so it means that many amino acids are coded for by multiple codons, 3 nitrogenous bases, but they code for the same amino acid all the time. They don't change, okay? If they were ambiguous, that would mean like, coming to later's class and one day an assignment's worth 1 point and one day an assignment's worth 5 points and one day assignment's worth 25 points. I don't do that because I'm not ambiguous. They're always worth 5 points. Okay, this is why, the genetic code. So there are 61 different codes for amino acids, and then the 3 are for stop codons, and a stop codon tells the RNA to detach from the ribosome. Okay, like we've learned before, the template strand is the strand of DNA that is converted to mRNA. It's also known sometimes as the lagging strand. It runs 3 to 5. That's because when the enzyme comes by, it's going to build the mRNA 5 to 3. RNA polymerase creates the mRNA once again in the 5' to 3' direction, just like DNA polymerase. All enzymes that deal with DNA. The non-template strand has the exact same basis as the mRNA, with the exception of uracil, okay? And it's called the coding strand. So I just put an example here, just showing you. Here's my template DNA, and it's 3' to 5'. And the one that runs 5 to 3 is called the coding strand, okay? And this is the mRNA right here. Here's my mRNA. So if you look at it, if I take the template strand, it's complementary to the template strand because T complementary to A complementary to U. Okay, but I like to go from the coding strand. If someone tells me it's the coding strand, and you'd get a lot of this in class, and especially on my tests and stuff too, it's real simple because all you do is go along that coding strand and you just go boom, A, U, G, C, A, C, A, U, A, U, G, A, G, U, G, A. Boom, right there. Okay, much easier to do, but hey, whatever you prefer. And then of course, we use our codon chart to figure out these codons. The first codon was figured out in 1968, and the first codon they figured out was phenylalanine, UUU. All right, but we use our codon chart. Notice, guys, that AUG codes for methionine, MET, and that's often we call the start codon. And then we have like histidine, lysine, tyrosine, glutamine. They usually end in INE, these amino acids. And then the last one is usually a stop, hopefully, okay, and that's where it's going to detach. If you didn't have a stop codon, it wouldn't detach from the ribosome, which means it wouldn't be built. And I was just saying here, note there's 18 nitrogenous bases here, 3, 6, 9, 12, 15, 18, and that's going to code, every 3 codes for an amino acid, so that's going to be 6 amino acids. All right, so methionine is your start. The last is called the stop. This is where mRNA detaches from the ribosome. All right, and then once that peptide is built, is now a polypeptide. Now it's going to go through those secondary and tertiary structures. The primary structure is just dictated by DNA, and that is going to be the order of amino acids. And then the secondary structure is where there's hydrogen bonding between the functional groups. So it's going to start to fold up into either a alpha helix or a beta pleated sheet, and then there's going to be the tertiary structure, and the tertiary structure, of course, is now more bonding amongst the R groups. And then if it's made of more than one polypeptide, that's where you're going to get to the quaternary structure. So under cracking the code, 61 was when the first amino acid was coded for. Okay, and then stuff we've already gone through. And guys, that is already the end of 17.1. Just one more quick word to say, and that is that the genetic code is 99.9% universal, and that means that you can put genes, in different organisms, and they're going to read it. So we often, it says, look at the picture of the smiley pig. There's a smiley pig. I think it's in your chapter, but that is using the jellyfish gene. And the jellyfish gene makes things glow. And they use that usually as a marker gene because when the organism is glowing in certain places, they know that certain genes are being read. Okay, so the next place we'll take this is we're going to go ahead and break down the steps. All right, and real quick, we're going through this because obviously you guys are aware that we have to get through some material here, and I'm just going to kind of highlight this stuff here, and this just basically breaks down transcription and talks about modification and then translation. Okay, and then we'll talk about at the end of the chapter, which is pretty simple actually, some genetic mutations. Okay, and genetic mutations would be when you change a nitrogenous base. What can happen? And that is 17. 17 is not a very difficult chapter in my opinion. It's kind of an interesting chapter, but it's very, to me, it's concrete. It makes a lot of sense. Once you go over it, it's okay. And then chapter 18, we're just going to hit the highlights too. But we'll start in chapter 18 with regulation. And that is, how does an organism know when to turn genes on and off? Okay, they don't just, they're responding to their environment. So very different, but you're going to learn more about that.